Chapter 4, A Tour of the Cell. One of the early books on cells was by Mr. Hook. Mr. Hook was one of the early people to publish on microscopes, but he was actually not the first person who discovered life under the microscope. That was Antony Leeuwenhoek. Antony Leeuwenhoek was a fascinating character. He was a citizen scientist. He was actually a guy who was selling carpets, and he made his own microscopes to see his carpets better, his fibers better. And then he got interested in looking at other things, and yeah, he found sperm, and like I said, he found life. And back in 1665, it was kind of difficult to convince other people of what you'd seen because they had barely invented microscopes. They hadn't invented cameras yet. I mean, film, the kind of things to record these things. So they were drawing these things. And then you had to say things like, well, I promise I saw this under the microscope. And you can imagine how that went over. Um, but it's a fascinating story, one of my favorites in science history. But we're going to look at just cells as we know them today. Again, the book gives you this division into major ideas. So we've got an introduction to the cell, and then we're going to look at the parts of the cell. We're going to look at the nucleus, the ribosomes, the endomembrane system, energy converting organelles, and then the cytoskeleton. So first, let's talk about the cell. Um, Again, the book starts with microscopes, and really the only two important terms that we need to think about here is magnification, which is the increase of the object. But there's also an important measure, that's the resolution. Um, Resolution is a measure of the clarity of an image. And you guys think of it as pixelation. If you have a very pixelated image, it doesn't matter how much bigger you make it, right? You cannot you cannot all of a sudden separate a pixel into 2 pixels because there's no 2 pixels. And that's the same thing. Resolution won't if the resolution isn't there, it won't allow you to separate 2 dots into 2 dots, meaning 1 pixel cannot become 2 pixels, so this thing doesn't become any sharper than it already is. um, Cell theory in the olden days, way in the olden days, they used to believe that there was this idea of life would just spring out of nowhere. And cell theory was the first time where it became clear that cells only come from cells. So that's like one of the very, very important things. All living creatures and things are composed of cells. All cells come from other cells. Cells are the smallest living things. Now, in terms of these smallest living things, they come in different sizes too. But just as a comparison, so and we need to get used to meters and nanometers and millimeters because they're so much more convenient than that stuff that you have to offer here. But a human height is about a meter 50 or so. So here's your meter. Then you've got the millimeter over here and the micrometer, that's your mitochondria, your nucleus. Your human egg is a very large cell, so this is what the light microscope can see. And then when we get smaller, we can see the electron microscope can still see this, but the smallest bacteria, then we've got viruses, and then finally atoms. So that's your size comparison here. Um, but what do you mean? What do we need for the cell? The very first thing is a plasma membrane, right? The plasma membrane is the thing that makes the cell, that gives the cell this compartment that it needs in order to be separate from the other things. Um, and there are channels that allow things to move back and forth, but this is the plasma membrane isolates the cell. The other thing we need to talk about is we need to talk about the size of cells. And when we're talking about cells have a surface area and an internal area, and the surface area and the internal area changes dramatically. The ratio between the two changes dramatically. So when you're looking at these two cubes, for example, you've got all of them have 27 units, right? Because you've got it's like 27 3 unit cubes. Total surface area here is 54. Total surface area here is 162. And so one of the things that we need to think about is for cells to function efficiently, um, they it really is only possible with processes that don't require energy, and diffusion is one of those processes. Diffusion, things just move, right, from a high concentration to a low concentration. And if something is small enough, diffusion is really very efficient. If you get too big, diffusion on long distances just isn't very efficient at all. And so small cells, like a bacterial cell, is small enough that it can rely on diffusion. A eukaryotic cell is, when you're thinking about the size comparison of a bacterial cell versus a eukaryotic cell, I did the calculation many years ago. Um, if you imagine a eukaryotic cell being about the prokaryotic cell being about the size of a washing machine, then the eukaryotic cell would be approximately the size of the stadium. Um, so you can see how if diffusion works in one, it doesn't necessarily work in the other. Um, so let's look at this membrane here for a second, just as a little reminder. When you look at the membrane, so you've got the phospholipid bilayer, and you've got the hydrophilic heads that are towards the water, and the hydrophobic tails that are sticking to each other. And then there are channel proteins that go through the membrane. Some of them are only on one side of the membrane, some of them are only on the other. And they're inserted in there, and the way they fit in there and the way they work in there is that the hydrophilic portions of the protein tend to be on the watery sides, versus the hydrophobic are inserted into the membrane itself. So let's see what we have in cells when we're looking at cells altogether. So the prokaryotic cells, which is a simpler cell, is bacteria and archaea. All other cells are considered eukaryotic cells. And eukaryotic cells, in contrast to prokaryotic cells, they do have a membrane enclosed nucleus and organelles to produce specific functions. Prokaryotic cells are smaller and simpler, but what all of them have in common, they all have a plasma membrane, they all have DNA, they all have ribosomes, and they all have a cytosol. Now, the prokaryotic cell looks sort of kind of like that. This is an example of a prokaryotic cell. You have the DNA is just part of the nucleoid over here. This is the chromosome, the one bacterial chromosome, which tends to be circular. There's a plasma membrane. Now, bacteria also have a cell wall a lot of times, and that's very characteristic for bacteria. And it's basically one compartment, right? Because bacteria are small. Now, a eukaryotic cell has to, because it's not so small, it has to make smaller compartments inside of there, and then diffusion can function within the smaller compartments, where some of the other function has to be handled in a different way. So these organelles that compartmentalize the activities of a eukaryotic cell are the nucleus and the ribosomes, which are the genetic control center. Organelles that involve the manufacturing, distribution, and breakdown of molecules, which include things like the endoplasmic reticulum, also called ER, Golgi apparatus, lysosomes, vacuoles, and peroxisomes. Then, and we're going to talk about these some more, specifically mitochondria, in all of cells, including plant cells and chloroplasts that are specifically in plant cells. And then we have components that are structural support components that have to do with movement, communication between cells, and those kinds of things. So your eukaryotic cell now looks very much more complicated than your prokaryotic cell, because it's, like I said, much bigger. Um, and in size comparison, the prokaryotic cell would be about the size of a mitochondrium. That's about the size comparison. But you've got the nucleus here, you've got the endoplasmic reticulum, and all of these membranes are interchangeable with each other. So, little vesicles pinch off here, um, and fuse with another membrane. So they all it's a very dynamic kind of thing. Plant cell looks very similar. The big difference between the plant cell and the animal cell is the plant cell has a cell wall that the animal cell does not have. The plant cell also has a central vacuole, which is very important for the trigger pressure that the plant cell needs in order to remain upright. Um, Nucleus and the ribosomes. Nucleus contains the DNA, and DNA, and we're going to talk about that in more detail in the future. But DNA is necessary. That's the blueprint of your of you of an organism, um, and it directs protein synthesis via messenger RNA, um, and the nucleolus is used to assemble subunits of ribosomes. So here's a picture of a nucleus and a nuclear envelope. It's usually a big nut-looking thing inside of the cell. Um, Ribosomes are made from ribosomal RNA, and they synthesize proteins according to the direction of the RNA. Now, cells that make a lot of proteins tend to have a large number of ribosomes. Those kinds of things, those form follows function. If you see that a cell or a structure has a lot of something, that's probably because it needs it. So here is a picture of the endoplasmic reticulum. And we have a rough ER and a smooth ER. The rough ER is the one where the ribosomes are bound, but there's also free ribosomes. The endomembrane system is a continuous system of organelles that interact in the synthesis, distribution, storage, and export of molecules. So there is the endoplasmic reticulum, which is basically a biosynthetic workshop. The smooth ER synthesizes lipids and processes toxins. The rough ER, which is the one that has the ribosomes on the outside, produces membranes, and the ribosomes make membrane and secretory proteins. Here's an example or a picture of the smooth ER and the rough ER. And here is an example of how this transport works. So you have a ribosome, that you have a growing polypeptide that gets turned into a complete glycoprotein. A transport vesicle pinches off, and then there are signal proteins on the outside of the vesicle that tell you where this vesicle needs to go to, and then it gets transported to wherever it needs to go. The Golgi apparatus is basically the post office that does these kinds of things. It contains it's a stack of membranes like that, and there's a one directional transport that happens from one side of the Golgi to the other side of the Golgi. Um, Mitochondria, as mentioned before, are in all cells, and chloroplasts are in plant cells for energy processing. And then we again have the cytoskeleton. Um, the other one that we need to talk about is the lysosome. They house enzymes that break down ingested substances and damaged organelles. Now, lysosomes, we got to be very careful with lysosomes, right? Because if lysosome contains digestive enzymes, and we are what we eat, right? This lysosome would digest anything. So if this lysosome opens up, it can digest just about anything there is. Um, so these lysosomes, they fuse with other vesicles to make sure that the digestive enzymes are very much contained within just the membrane. Vacuoles um, exist in some protists. Protists have contractile vacuoles a lot of times in order to make sure that the water that enters the protist at all times gets kicked back out. Um, plants often contains Plant cells contain a central vacuum that stores molecules and wastes and that kind of thing. Um, Here's an example of a contractile vacuole um, on a paramecium. And a paramecium, because and we're going to talk about that as part of osmosis in a minute, or in the next chapter, um, because there's more stuff inside of the cell. Water constantly enters the cell, and so the paramecium constantly has to push water out through this contractile vacuole. And then here's an example of a central vacuole in the case of a plant. Now, in this next slide, you see how these different structures are connected with each other. So you've got the nucleus, you've got the smooth ER, you've got the rough ER right there, and then the transport vesicles move from the rough ER to the Golgi apparatus, where they're transported some more. Um, there are connections between the Golgi apparatus and lysosomes. Um, and then the other one we've got is peroxisomes, which are other metabolic departments that don't originate from the membrane system. Now, next is the energy converting organelles, and we're going to talk about those in more detail later. But basically, what we have here, we've got the mitochondria, which have two internal compartments, and there's an intermembrane space. Now, this is very important. Um, because it tells us a little bit of something about the origin of the mitochondria. The mitochondrial matrix contains mitochondrial DNA, which is different from our DNA. We don't actually encode for mitochondria. It contains ribosomes and a lot of the enzymes that catalyze some of the reactions for cellular respiration. We're going to talk about cellular respiration in one of the future weeks. This is an example of a mitochondrium. And like I said before, it's about the size of a bacterium. Um, the other organism, the other organelle that's kind of like a mitochondrium is the chloroplast. It's the site of photosynthesis. This is what a chloroplast looks like. It also has a double membrane. It's also about the size of a bacterium. And it also has these stacks of membranes. Um, it is, we are pretty sure that what happened, and this is the endosymbionts theory, that mitochondria and chloroplasts were formerly prokaryotes that lived their lives outside of a larger cell initially. But then at some point in their lifetime, they were engulfed um, by a eukaryote that didn't eat them. And they discovered this symbiotic, or they established this symbiotic relationship that was a benefit for both of them. And the thought is that the mitochondrium came first because everybody has mitochondria, but only some have chloroplasts. So the mitochondrium came first, and then cells can do photosynthesis, but they have mitochondria too. Next is we got to talk about the cytoskeleton and cell surfaces a little bit. We used to think that cells were basically these bags of water, but they're not. There's tons of stuff going on inside of a cell. And the cytoskeleton includes things like microfilaments, intermediate filaments, microtubules, and they maintain the cell shape. They help with movement of things within the cell, and they also help with contraction. And so here are examples. So this is what a microtubule would look like. This is what an intermediate filament looks like, and this is what microfilaments look like. I'm not going to talk too much about this. Here's a picture of I'm going to talk a little bit about how cells move. There's two different ways. There's cilia and flagella. Flagella tend to be, let me show a picture of these are cilia. They tend to be shorter, they tend to work in waves, and flagella tend to be longer. They're more like whip-like. And that's how they work. The way they function is there is this structure, this microtubule structure, and the way this thing moves is by having these cross-linking proteins move against each other. And there are animations that you can see that show you how this happens. The other part that's of interest is the extracellular matrix. The extracellular matrix is on the outside of the cell. The main function of the extracellular matrix is things like transport, recognition, connection to other things. There's a lot of regulation that happens between cells, right? Some of that regulation is controlled and determined by different cell junctions. We've got three different ones we did distinguish: tight junctions, anchoring junctions, and gap junctions. There's a picture of these. Tight junctions basically make things tight. They make sure that there's nothing that can go between anything. Anchoring junctions connect things together, and gap junctions allow the loud distribution of molecules or whatever between between cells.